Hi everyone! So today while I was having dinner, a thought just clicked in my mind. I started thinking, we all love eating food of course, right? So what happens to the food that we consume after it's been digested and absorbed in our bodies? Well, today I'll be getting behind the physiology of this. So hopefully by the end of my presentation, all your questions will be answered regarding what happens to the food that we consumed after it's been digested and absorbed in our bodies. So keep watching. Wait, but before we start the presentation, we'll just go over what digestion and absorption is, just for our general knowledge, so it's easy for us to understand the presentation, and then we'll move on to the physiology behind digestion and absorption. So digestion is basically when the food that we've consumed is broken down into smaller building blocks, and this could be achieved by either chemical means such as chewing or churning, or mechanical means such as digestive enzymes of stomach and intestine. Absorption, on the other hand, is when the digested building blocks are absorbed into the bloodstream or lymphatic systems through the intestines and then transported to various cells, tissues, and organs in the body. Now that we have a general idea of what digestion and absorption is, let's move on to the different types of digestion and absorptions in the presentation. So in this presentation, I'll be discussing the digestion and absorption of carbohydrates, proteins, and lipids. So let's start off with the digestion of carbohydrates. The chemical digestion of starches begins in the oral cavity, which is the mouth. It then moves down into the internal organs and ends at your anus. So in the small intestine, the pancreatic amylase basically does the heavy lifting for starch and carbohydrate digestion, as seen in this diagram here. After amylase breaks down starches into smaller fragments, the brush border enzyme alpha-dextrinase starts working on alpha-dextrin, breaking off one glucose unit at a time. So the starch glycogen breaks down into disaccharides, and it hydrolyzes the maltose, sucrose, and lactose into monosaccharides. So what that basically means is that maltase breaks down maltose into 2 molecules of glucose, sucrase breaks down sucrose into 1 molecule of glucose and 1 molecule of fructose, and lactase breaks down lactose into 1 molecule of glucose and 1 molecule of galactose. And keep in mind that insufficient lactase can lead to lactose intolerance. Now let's move on to the absorption of carbohydrates. As we all know, all carbohydrates are absorbed in the form of monosaccharides, and the small intestine is highly efficient at this, absorbing monosaccharides at an estimated rate of around 120 grams per hour. All normally digested dietary carbohydrates are normally absorbed, and indigestible fibers are eliminated in the feces. The monosaccharide fructose, which is usually seen in fruits, of course, is absorbed and transported in facilitated diffusion alone. And the monosaccharides, on the other hand, are combined with the transfer proteins immediately after the disaccharide is broken down. Now let's move on to the digestion of proteins. We consume around 15 to 20% of our daily calorie intakes as proteins, and proteins are basically polymers which are composed of amino acids linked by peptide bonds, and they form long chains. So digestion basically reduces them to the constituent amino acids, and the digestion of protein starts in the stomach where hyaluronic chloride and pepsin breaks down proteins into large peptides, and then they break into short peptides and amino acids, and then they finally break into amino acids where they're able to travel into the small intestines. Chemical digestion in the small intestine is continued by pancreatic amylase, which include chymotrypsin and tritrypsin, each of which act on specific bonds in chemical amino acid sequences. At the same time, the cells of the brush border secrete enzymes such as aminopeptidase and dipeptidase, which further break down peptide chains. This results into the molecules to become small enough that they are able to enter the bloodstream. Now let's move forward to the absorption of proteins. The active transfer mechanism, primarily in the duodenum and the jejunum, absorb most proteins as they break down products amino acids. Almost around 95 to 98% of proteins are digested and absorbed in the small intestine, and the type of carrier that transports an amino acid varies. Most carriers are linked to the active transport of sodium, and short chains of 2 amino acids, which are known as dipeptides, and 3 amino acids, which are known as tripeptides, are also transported actively. However, after they enter the absorptive epithelial cell, they are broken down into the amino acids before leaving the cell and entering the capillary blood via diffusion. Now let's move on to the digestion of lipids. A healthy diet generally requires us to have around 35% of our total calorie intake as lipids. The three lipases responsible for the digestion of lipids are lingual lipase, gastrolipase, and pancreatic triglyceride lipase. However, because the pancreas is the only significant source of lipase, virtually all lipids digestion occurs in the small intestine. Pancreatic lipase breaks down each triglyceride into two free fatty acids and a monoglyceride. The fatty acid includes both short chains, which is less than around 10 to 12 carbons, and a long chain fatty acid. Lastly, we've got the absorption of lipids. So around 95% of lipids are absorbed in the small intestine. As seen on this diagram, the fatty acids, which are relatively water-soluble, and monoglycerides are emulsified by bile salts to form micelles. And the bile salts not only speed up the lipid digestion, but they also are essential to the absorption of the end products of lipid digestion. Next, we move to the fatty acid, which enters the epithelial cell and links to form triglycerides. And then moving on to the triglycerides combined with the proteins inside the Golgi body to form chylomicrons, which are a small fat globule composed of protein and lipid. And then we move to the final step, which is chylomicrons enter the lacteal, which is basically the lymphatic vessels of the small intestines, which absorb digestive fats and are transported away from the intestines.